I'm a physician. Went to medical school at NYU in New York, and then moved out west, did my residency in pediatrics before returning to the National Cancer Institute in Washington, D.C., or Bethesda, Maryland, where I spent 4 years as a clinical associate, uh, and then moved out to a faculty appointment in the clinical department at the University of Utah. And it was there that I noticed two things. The first was that many of the clinical faculty in the department, I noticed, were working extraordinarily hard, seeing patients, teaching medical students, doing all the BS that's part of being a university faculty member, and in their spare time trying to do research. And quite frankly, only a few people managed to do a good job of any of those. And I knew I wasn't one of those remarkable individuals. And I knew also that what I was most interested in was research. And so my career took a kind of a left turn. And it was influenced by actually a scientific discovery. I was a pediatric rheumatologist taking care of kids with autoimmune diseases, and it was at this time that Lyme disease, which had been diagnosed in 10 kids as juvenile rheumatoid arthritis, classic juvenile rheumatoid arthritis, turned out to be an infectious disease. And I began to worry that, gee, maybe all of these inflammatory diseases that we were involved in were in fact infectious, and here we were treating them by immunosuppression. And so, as I said, my career took a left turn. I moved to an NIH laboratory located in Hamilton, Montana, which is actually where the Lyme disease spirochete was discovered. It was originally just supposed to be a sabbatical year, but I spent 8 years there and really never ever came back to clinical medicine. I was there for 8 years. That was the time when HIV was becoming of interest, and I actually thought that studying HIV would bring me back to autoimmune disease because I thought retroviruses might in fact be responsible for many autoimmune diseases, and that's still a viable hypothesis. And so I can't look both at the camera and what I'm reading, so you're just going to have to see the side of my face. Anyway, moved out to, after working at the Rocky Mountain Labs, moved to across the state of Montana to Bozeman, Montana, where I was head of the Department of Microbiology, and then eventually down to New Orleans, where I've been the director of the Research Institute for Children here and a faculty member at LSU Health Sciences Center, and have been involved in a number of professional committees and what have you. So it's a slightly different career arc. Started out in clinical medicine, but really knew even when I was a medical student that I wanted to do research, and so aimed towards that. I've had administrative responsibilities along the way, and that's fairly typical. Okay, next slide, please. So, what do we do in my lab? I'm interested in antibodies. I find these fascinating molecules, and it's been a great time to be interested in antibodies. We know that they're important components of vaccine protection. Now we're in an age of therapeutic antibodies. In fact, 7 of the top 10 selling drugs in the United States are therapeutic monoclonal antibodies. And they're extremely useful because they also allow us to do immunoassays, which can be developed to measure rather simply any number of different substances. They're used throughout research and medicine, but also in industrial processes and environmental monitoring as well. So antibodies, to me, I sort of joke, they're a molecular monkey wrench. They have an antigen binding site that can be tailored quite specifically to bind almost any molecule you can think of. And then they have a tail end that contains constant domains that actually cause effects. They're the part of the molecule that causes a biological effect. And these effects can be profound. They can be used to treat cancer, prevent infectious disease, and so forth. So what do I do with antibodies in my laboratory? Next slide. Okay, what we're trying to do, to put it quite simply, one is to cure HIV infection, which involves eliminating the latent reservoir of the virus, and we'll talk about that. I use both antibodies and antibodies that have been coupled to poisons to target these HIV-infected cells. We're also working in the bioterrorism arena. Ricin is a common toxin that we worry about. The ricin plant is the castor bean plant. It grows both ornamentally and as a weed throughout the world. The toxin is easy to purify, and a milligram of that toxin is enough to kill a person. And we're trying to develop treatments post-exposure. And then finally, we're in both of these experimental systems, we're engineering better antibodies, or trying to do so. And that's what our summer students are working on. So, talk a little bit about HIV. We have had effective therapies for HIV infection for well over a decade now. But when we look and see the decay rates of the latent reservoir of HIV, okay, so when HIV infects a cell, the virus integrates into the cell's genome and hangs around as long as the cell does. It infects long-lived memory cells, and immunologic memory lasts a lifetime, and so do those cells. And so the decay rate, even with effective antiretroviral therapy, is extremely slow. And it's been estimated that it would take greater than 70 years of treatment to eliminate the latent reservoir of HIV that would allow people to then stop taking these drugs. And so what we're trying to do is hurry up that process. Next slide. And we're taking two different approaches. The first is what we call subset targeting. And so that's to identify the cells that carry this HIV virus around in their genome and eliminate them. And as I said, long-lived memory cells are certainly one subset that we want to eliminate. And also we think that macrophage lineage cells, which are also long-lived, may carry the latent virus. And so we use cell sorting to identify the cells that carry the virus. And we've actually done CD4 depletion experiments in an animal model that we've developed in an effort to eliminate those cells that might carry the latent HIV. So that's one approach, is subset targeting. The second approach, go ahead, is to use what we call anti-HIV immunotoxins. So we want to kill cells that actually express HIV. So in fact, the HIV envelope protein, which is called GP120 and GP41, is expressed on the surface of the virion, but also on the surface of cells that are infected. So as the virus buds out from the cell, which you can see in the right-hand picture, the cell itself carries the HIV envelope. And so we've taken ricin toxin, which is actually how I got to study ricin, I wasn't initially interested in it as a bioterrorism agent. I was interested in it as a therapeutic. And we take the toxic part of the ricin and couple it to an anti-HIV antibody, or alternatively, we can take a small drug and conjugate it to an anti-HIV antibody. And the next slide shows you what happens to cells exposed to these toxins. So in panel A, you can see what a normal lymphocyte looks like in close-up view. This is a scanning electron microscopy picture at about 20,000 magnification. The infected cell, you can see the viruses budding out on the surface of the cell. And then a half hour after we expose the cell to this ricin immunotoxin, you can see the big gaping holes in the cell surface and how we've killed the cell. And so it works well, but we needed an animal model to test this in. And that's where we've been putting most of our efforts. And we're working with a chimeric virus called SHIV. It's a mixture of HIV and the monkey equivalent SIV. It was developed for vaccine work, and it's turned out to be sort of a dud in terms of vaccine work because it'll infect the animals, but then the animals seem to clear the infection. But in point of fact, they don't clear the infection. If you look at the graphs on the left side, you see that both rhesus and pig-tailed macaques, two different species of macaques, if we look at the number of cells in their circulation that carry the SHIV virus, it's somewhere between 10 and 1,000 per million cells that are circulating even years after the animals have cleared the virus infection. And so, in fact, it sets up a latent infection. And on the right side, we show that these animals, we can activate these animals or activate the latent infection by a maneuver called CD8 depletion. We get rid of the killer T cells that seem to be controlling the virus. And so what we find is that these animals in the red have a very high peak of viremia that goes on for about 2 to 3 weeks, actually higher levels of virus than initially. So these animals actually do have a latent infection even after years of no treatment. And so we're able, as I showed 2 slides before, to test these different approaches in these animals, and hopefully we'll come up with an approach that will allow us to eliminate this latent reservoir of HIV, which is really the barrier to curing HIV infection. And so one of the key things is to find the very best antibody for targeting these cells. And so on the next slide, I just summarize getting better antibodies. And what we're doing is we're doing an approach called double variable domain antibodies. So on the top of this figure, we show the HIV envelope protein in its 3-dimensional structure. It's actually a trimer of GP120, GP41s. So there are 3 GP41s, 3 GP120s. And what we've done is we've taken an antibody that binds to GP120 to a conserved site, and another antibody that binds to GP41. And you can see the binding sites of those antibodies in purple and red on that trimer molecule. And we've made what's called a double variable domain antibody that can bind to both parts of the HIV envelope, not just one part. And we're now testing these antibodies. And as I said, this is a project that several of our summer students are involved in. And so I think I'm going to stop there. Did I stop there? Is that my last slide? Yep, I stopped there. And so if any of you have any questions, I can answer them.
Thank you. Nice presentation.
Okay, I'm not seeing any hands. If anyone wants to
Any
yell or
questions?
speak out,
Any questions here?
otherwise, I guess we're finished.
Any questions from the Health Science Center New Orleans? Any questions from Health Science Center in Shreveport? Everyone's very quiet.
Well, thank you, Dr. Pincus, for presenting for us today.
Okay.